So for today's notes, we're going to look more specifically at how the process of cellular respiration actually occurs inside the cell. So the first thing to do is to label our picture here of an organelle called the mitochondria. And we want to label it with where our steps of cellular respiration actually occur. The first stage of cellular respiration is called glycolysis, and that occurs right here in the cytoplasm of the cell. So that's stage number 1. So before we even enter the mitochondria, glycolysis, the first part of cellular respiration, is occurring. Then stage number 2 is called the Krebs cycle, which occurs right here. You can tell it looks like a cycle with the arrows. And then stage number 3 is the electron transport chain, which also occurs inside the mitochondria right over here. So which stage occurs in the cytoplasm? Well, the only one to occur in the cytoplasm would be glycolysis. And then these last 2 stages of cellular respiration, Krebs cycle and the electron transport chain, both occur in the mitochondria. Now that we have an idea of where things are happening, let's actually look at the specifics of what is happening, what the biochemistry is of this breakdown of glucose into energy. So the first stage, glycolysis, is going to start with the reactants, 1 molecule of glucose and 2 molecules of ATP. It's going to end up producing 2 pyruvate molecules, which is a type of carbon compound, and 4 ATP molecules. So the goal here is to get some energy from the process of glycolysis, but notice how you have to spend energy in this case in order to get energy. Ever hear you have to spend money in order to make money? In this case, the process of glycolysis, in order for it to get started, actually spends 2 ATP. So -2 ATP is spent, but it's going to make 4 ATP. So with that, what would be the net gain of ATP molecules? Think about that for a second. -2 plus 4. If you guessed 2 ATP, then you are correct. 2 ATP are gained from the process of glycolysis. Also produced, there we go, 2 ATP. Also produced by this process is a molecule of NADH. So 2 molecules of NADH are made. This is another electron carrier, looks similar to one we learned about for photosynthesis called NADP, right? P stands for photosynthesis, H. This is a similar electron carrier like a battery that is going to be charged up by this process as well. Because glycolysis is the first stage of cellular respiration. It is not the big energy making stage. It is preparing glucose into the pyruvate molecules, which are molecules that are going to be able to continue the rest of the process and generate more energy. And this electron carrier is also going to later be used to generate more energy. So it's a bunch of preparation to get the cell ready. Some advantages of glycolysis is that it can happen very quickly. Cells can produce thousands of ATP molecules in a few milliseconds, even though they're only generating 2 ATP molecules for every 1 time it goes through glycolysis. But the other most important advantage of glycolysis is that it does not require oxygen. Because it does not require oxygen, we consider this an anaerobic process. There are times in a cell's life, which we're going to touch on more in a later topic, where oxygen is just not readily available, but the cell still needs energy. So being able to do glycolysis without oxygen is very important for the cell. But that's still, again, not our biggest energy maker. And the point of cellular respiration is to make a lot of energy. So when oxygen is present, this process from glycolysis moves on to the next stage of cellular respiration, which is called the Krebs cycle. So oxygen will be required for the final steps of cellular respiration, the Krebs cycle, and the electron transport chain. Because of that, we consider them aerobic. Good vocab word. So during the Krebs cycle, pyruvic acid is broken down into carbon dioxide in a series of, see that word, energy, the whole point of this, energy extracting reactions. So the pyruvic acid that came over from glycolysis is what is going to be broken down in order to extract energy. And carbon dioxide is the main product of this besides the energy, and that is what we are going to exhale as a waste product of our cells, is carbon dioxide. Well, how does it actually do this? So a little bit more detail into the steps of the Krebs cycle. It starts when pyruvic acid produced by glycolysis enters the mitochondria. Remember, glycolysis happened in the cytoplasm, so this was outside of the mitochondria, the energy factory for the cell. So pyruvic acid over here has to enter in to the mitochondria, step number 1. Step number 2, once pyruvic acid has entered in, one carbon molecule is removed from it, forming carbon dioxide. And the electrons that were being used to hold on that carbon molecule together are also going to be removed, and it's going to charge a molecule of NAD plus into a molecule of NADH. So carbon dioxide and NADH are produced. Then what is left over, the carbon molecule that's left over from that change, is going to join with an enzyme called coenzyme A, and that forms a molecule called acetyl-CoA. You can see that here in step number 3, acetyl-CoA. Oh, that's not a very good color, right here. Step number 4, acetyl-CoA, that molecule, is now going to add 2 more carbons to a 4-carbon compound that is already part of the cycle. And that is why sometimes this cycle is called the citric acid cycle. That is the name of the compound that is present in the cycle. So it is known as the Krebs cycle, or aka the citric acid cycle, because of this molecule right here that is going to pick up these acetyl-CoA molecule. Number 5, that citric acid is now going to be broken down into a 5-carbon compound and then into a 4-carbon compound. So carbon is constantly being taken away from these molecules, and in doing so, it's charging up our energy carriers. But it's also making that carbon molecule smaller as it goes around the cycle. 2 more molecules of carbon dioxide are released in this process. Carbon's got to go somewhere, and it joins up with the oxygen and is released from the cell. And those electrons, from taking that carbon off, it releases energy in the form of electrons, and those electrons form NADH and another electron carrier unique to cellular respiration called FADH2. Both of those are like our batteries. Got NADH is like our on-brand battery. FADH, which is less common, is like our off-brand. But regardless, step number 6 is about charging up these energy carriers that you can see throughout the cycle here are being produced. And step number 7, 1 molecule of ATP is generated. So all that work for just one molecule of ATP. You might be thinking, wait a minute, glycolysis gave us 2 molecules of ATP. That at least is a little bit better than this one. But do remember that this is only for one molecule. Oops. This is only for one molecule of pyruvic acid. And remember, glycolysis generates 2 molecules of pyruvic acid from one molecule of glucose. So technically, this cycle has to spin 2 times in order to get the energy from one molecule of glucose. So technically, 2 molecules of ATP could be generated from one molecule of glucose if the Krebs cycle spins around those 2 times. So now we can add up our energy totals that we get from the Krebs cycle from just one molecule of glucose. So 2 times around with the Krebs cycle for each molecule of glucose is going to give us the following totals for energy. So we're going to be left with 8 NADH, 2 FADH2, and 2 ATP. Because each turn around the Krebs cycle is only accounting for half a molecule of glucose. So we have to turn it twice in order to get the full molecule of glucose and to generate these totals. So 8 NADH, 2 FADH2, those are our electron carriers, and just 2 ATP. So at this point, we really only generated 4 ATP total, 2 from glycolysis and 2 from the Krebs cycle. The highest amount of ATP or energy generated from cellular respiration is going to be in the 3rd and final stage of cellular respiration, and that is the electron transport chain. So in addition to the ATP that's been generated, these energy carriers, these electron transporters, have been gathering those electrons that they're going to transport to the electron transport chain to actually generate the most energy. So this 3rd step, the electron transport chain, uses the high energy electrons from the Krebs cycle and a little bit from glycolysis to convert ADP. Remember, ADP is the low battery form of ATP. And they're going to convert it into that high energy form of ATP that the cell will be able to use for energy. So they're taking the electrons from the previous 2 steps, and this transport chain will use those electrons to generate molecules of ATP that the cell can use for energy. So how does this happen? Well, the first step, the high energy electrons from those 2 molecules, NADH and FADH2, are going to be passed along this electron transport chain from one carrier protein to the next. There's these little blue proteins actually inside the membrane of the mitochondria, and they kind of act like skipping stones. So as each electron bounces down here, they're going to be generating some energy. Now the electrons need somewhere to go though. So at the end of this little chain, like when you're skipping a stone, what happens? The stone eventually falls into the water, right? If you're skipping a stone across a pond, eventually it falls into the water. Well, that also happens with the electrons. At the end of the chain, water is going to be formed by those leftover electrons. So water will be formed, and the energy used as those electrons bounce down is going to be used to generate ATP. So these electrons bounce down, they cause hydrogen to bind with oxygen, which creates our water molecules, and then the extra energy from those electrons is going to be used to move hydrogen ions across the membrane. So this is how the ATP will be generated. So as those electrons bounce down, the energy they release moves these hydrogen ions. I'll use a little bit thicker of a pen here. Across the cell membrane. Once they're on this side, they're like, wait a minute, we didn't want to come over here. We have to get back to the other side. This is called concentration gradient, which we'll learn about in our cell transport section. So once these hydrogen ions are moved across the membrane because of those electrons, they actually want to come back into the cell. So the mitochondria. So step number 4 actually has those hydrogen ions escaping back through the membrane channels, causing this molecule right here, called ATP synthase, to spin, kind of like a water wheel or a wind turbine. So these hydrogen ions got pushed across the membrane, and they're like, wait a minute, we don't want to be on that side. So they're all going to come over here, and they're going to enter back in the only way they can, and that's through this little tube of ATP synthase. And as they come back through, this tube turns, and that spinning motion generates a molecule of ATP. Oh, we should highlight that too. So fill in, generates a molecule of ATP as the ATP synthase spins. Every single electron carrier has to do this. So we've got our NADH and our FADH, all the totals that we made, dropping off all their electrons. As that ATP synthase is spinning, it's going to make the most amount of ATP. So if we add it all up at the end, our totals from one molecule of glucose for cellular respiration is 2 ATP from the glycolysis, 2 ATP from the Krebs cycle, 32 ATP from all those electrons in the electron transport chain, which gives us a total of 36 ATP from one molecule of glucose. Now, this isn't an ideal world or an ideal cell. Not every cell or every mitochondria in a cell is perfectly efficient at generating this amount of ATP. Some actually perform better and generate more ATP. Some don't do as well and they generate less. But the number that I want you to remember, and kind of the number that we're going with here, is this breakdown. So 2 ATP from glycolysis, 2 ATP from the Krebs cycle, 32 ATP from the electron transport chain, for a total of 36 ATP from one molecule of glucose. And that is a wrap-up of the specific steps of cellular